Okay, I'm continuing the subject of insulin and the function of the insulin hormone. At this file that we are talking about the function of insulin, you are comparing the ratio of glucagon to insulin, glucagon plasma, and the glucose concentration in the course of time. Imagine we increase the blood glucose from the time zero here. Where is the here to very high levels. This time glucose is increasing. As you see, at the same time, immediately insulin is also increasing, and with a delay, glucagon start to decrease, and glucagon also increase, and then start to decrease. But you may say that glucagon is increasing, insulin is also increasing. They have a discrepancy between these two effects. It is not, because when you have a look at the glucagon to insulin ratio, glucagon to insulin ratio is dropping, even though glucagon is decreasing and insulin also, but insulin is increasing more intensely. Therefore, the glucagon to insulin ratio was decreasing. What will happen? This will cause the glucose concentration to decrease. And as glucose concentration is decreasing, the ratio of glucagon to insulin is increasing. And at this point, even though that glucagon was decreasing, but insulin was more pronouncedly decreased. Therefore, at this time, we have a high glucagon to insulin ratio. It means that glucagon is predominant. And this will stabilize the blood glucose. Do not allow the blood glucose to drop forever. It will be smooth and there remain at its normal level. This remaining of glucose at its normal level and do not allowing it to drop is because of this play that we have between insulin and glucagon and the ratio of glucagon to insulin. Have a look at the liver glycogen content. Over time, the glycogen content of liver is increasing. It means that this high level of glucose and by the help of insulin is converting into glycogen, and the storage of glycogen is increasing. And at the same time, the glucose uptake, the hepatic glucose release will be decreasing because we do not need glucose from the hepatic cell to be released. We have extra glucose available in the blood. Therefore, it will drop. And this drop is simultaneous with is in accordance with the increasing glycogen storage. But over time, that glucose concentration is decreasing, and we want to maintain the glucose. This time, this high glucagon level stimulate the hepatic release of glucagon, and the hepatic release of glucagon is increasing. And on the other hand, liver glycogen content is decreasing. Therefore, it shows that these glycogens are being released as glucose in the blood. This figure shows the source of blood glucose during a normal day. And the blue line shows the source of dietary glucose. And as you see, after each meal, breakfast, lunch, and dinner, every time that we eat the food, immediately the source of and the role of the dietary glucose will increase. But over time, and then on the other hand, the role of gluconeogenesis and glucogenolysis, the release of glucose from other source and storage forms will decrease. But over time, from breakfast to lunchtime, you can see that gradually the glucose that is absorbed from the GI system is decreasing. What will happen then? Body need glucose, and to not allowing this to drop the glucose concentration. As you see, the green line show the glycogenolysis. Those glucose that were stored in the liver and the muscle will be released as glucose, and this causes the glucose to remain, and therefore the source of glycogenolysis will increase. And if this was not able to restore the glucose to its normal and hold the glucose to its normal level, then another mechanism also will help, and this is glyconeogenesis, formation of glucose from other sources such as amino acid and fatty acid. Again, at the time of launch, immediately these two sources will drop to zero, and again the source of food is important, and another cycle of glycogenolysis and glyconeogenesis, and another cycle of eating behavior. And you know that during the night time, that is a long fasting period, you can see that firstly glycogenolysis is very high, but then during the night time, the glyconeogenesis, formation of glucose from the amino acid and fatty acid, will be the dominant form compared with the daytime. The glyconeogenesis during the daytime is not very significant, but during the night time that we are sleeping and do not eat anything, this do not allow the blood glucose to drop during the night time. And this show the level of glucose that after each meal it is increasing, and then because of the hormone is decreasing, and then for example here is increasing. Here we have another food, it is increased, and then because of the glucagon and insulin, as I told you, the ratio of glucagon and insulin, the glucose will decrease, and then another increase and decrease. And as you can see, the insulin show the most oscillation during these meal and fasting periods, and glucagon show lower change, just changing the ratio of glucagon to insulin. The next item that we have in our food is fat, and for fat metabolism, the adipose tissue is the most important part. Glucose transport into the adipose tissue is stimulated by insulin. Therefore, we say that insulin stimulates the glucose uptake by adipose tissue. On the other hand, when we have lots of glucose, there is no need for fatty acid to be used as energy. Insulin decreases the utilization of fat as energy source and decrease the release of fatty acid from the adipose tissue because it inhibits the lipase enzyme. Therefore, we say it favors to a storage of fatty acids. Not only do not use the fatty acid as energy source, but also convert some glucose into the fatty acids. The net result is fat deposition, lipogenesis. In the liver, the glucose transport into the cell will happen, and this will be converted into fatty acid and fatty acid synthesis and increasing fatty acid, increasing triglyceride, and increasing LDL. The net result of insulin in the liver during the feeding period is lipogenesis. And you know that for this reason, if we have insulin deficiency, the fat will be used more frequently for energy source because insulin is not available to inhibit it. Glucose is not used, therefore we have hyperglycemia and fatty acid will be used. Lipolysis of the lipid in the adipose tissue will increase because insulin is not available to inhibit this process. Fatty acid will be released into the blood, cholesterol will increase, phospholipid will increase, and therefore we have metabolic signs of increased lipid profile in the diabetic patients. And sometimes this may cause acidosis, ketosis, and even to the level that is dangerous for the body. This shows how much the absence of insulin will increase the blood glucose. At the same time, it also increases the fatty acid, free fatty acid levels. And at the same time, the acetoacetic acids, and for this reason diabetic affect the lipid metabolism. Finally, we have protein metabolism. What is the role of insulin in protein metabolism? I told you that insulin prevent protein to be used as energy source, prevent conversion of amino acid to glucose, inhibit gluconeogenesis. It stimulates the transport of amino acid into the cell, not for using as energy source, but for using them to produce new proteins. Therefore, we can say that insulin stimulates protein synthesis, increase translation of messenger RNA, increase the rate of transcription of selected DNAs, inhibit catabolism of protein, and in the liver, insulin inhibit the gluconeogenesis. All these effects is favoring growth. Without insulin, growth will be retarded. This figure clearly shows that if we remove the pancreas from the body of one animal that has a normal growth, immediately the growth will stop. If we remove the pancreas and the pituitary gland, therefore we have no growth hormone, no insulin, the growth will completely stop. If we inject the growth hormone without insulin, you can see that here we do not see any increase in the growth. If we inject insulin alone without growth hormone, it is not able to increase the growth. I want to say that the growth promoting effect of insulin is dependent on other hormones such as growth hormone. As the same way that growth hormone is promoting functions is dependent on insulin. And when we inject growth hormone and insulin together, you can see that the speed of growth return to its normal level. This shows the level of the dependency of growth on insulin, and also other hormones such as thyroid hormone that I told you before, such as growth hormone that I told you before. And this figure summarizes the metabolic function of the insulin, that you can see glucose is uptake into the muscles, amino acid is higher, amino acid entry into the muscle is higher than uptake of amino acids. Therefore, we have net result of growth. In the liver, we see that glucose release from the liver is decreasing. Amino acid uptake and converting into glucose is decreasing, is inhibited. In the adipose tissue, glucose uptake is increased and it is converted into the glycerol. On the other hand, release of glycerol and fatty acid will decrease. Instead, fatty acids are being absorbed and stored in the adipose tissue. And this figure shows the summary. And as you can see, glucose uptake by the brain is insulin independent because it is by the GLUT3. In the liver, glucose uptake is insulin independent because it is uptake by GLUT number 2. But in the adipose tissue and muscle cell, it is insulin dependent because it is GLUT4. Okay. This table show you the summary of the result that I told you in various tissues such as adipose tissue, muscle, liver, and in general. Please read these items that I told you before, and I am not going to repeat them. The next hormone, that was all about the insulin. The next hormone that is released by the pancreatic islet is glucagon. As I told you, glucagon is the opposing factor for the insulin, and it do a push and pull mechanism for insulin. It is an unbranched protein chain that has just 29 amino acids. The molecular weight is very low, and its gene is expressed in the chromosome number 2. It is expressed in the alpha cell but also it is also expressed in other cells, such as in the some area of the brain cells, some intestinal epithelial cells, and it has a lifetime of half-life of 5 minutes. What is the role of glucagon? I can say that all the effect that we know about insulin will be converted by the and reversed by the glucagon. Glucagon increase glucogenesis, increase gluconeogenesis, formation of new glucose, increase glucogenolysis, and increase free fatty acid secretion. It is this item is increasing glucogenolysis. Also, it has some other non-metabolic effect. For example, we know that on the heart, it has increasing function on the strength of the heart. It increases blood flow to the kidney. It increases bile secretion. It inhibits gastric acid secretion. And when we want to evaluate the glucagon secretion, again, we have some stimulatory factor, some inhibitory factor. Amino acids, acetylcholine, norepinephrine and epinephrine, GIP, and CCK stimulate the alpha cells. On the other hand, the glucose, insulin, somatostatin, ketones, GLP-1 inhibit glucagon secretion. The most important factor that stimulates glucagon secretion is reduction and lowering of glucose concentration in the blood. Every time that glucose concentration in the blood is decreasing, glucagon will be stimulated. And on the other hand, blood amino acid increase in the blood in amino acid will stimulate. One factor that stimulates the glucagon secretion is exercise. Do you know why? Because exercise was stimulating the uptake of glucose, therefore decreasing the glucose concentration. And this is showing the ratio, the balance that we have between the insulin different states. For in the fed state, because we have high glucose available, insulin will dominate. We have higher glucose oxidation, higher glycogen synthesis, higher fat deposition, and higher protein synthesis. But during the fasting state, glucagon will be dominated. Therefore, gluconeogenesis will increase, glucogenolysis will increase, and ketogenesis also will increase. And insulin effect will be a little decreased. These were the main hormones that I was going to discuss in the pancreatic isolate. As I told you, we have some other hormones such as amylin, such as somatostatin, such as these hormones are also important. But because of your time and your curriculum that we have not enough time, I prefer to limit this topic just to these two main hormones and the function of these hormones. And when we are talking about insulin and glucagon, you know that the most well-known metabolic disease is diabetes, and diabetes refers to the insulin and function of insulin. You know that we have two types of diabetes. One is insulin dependent, or type 1, that in this situation, the release of insulin is inhibited or is impaired. We have shortage of insulin. But there is another type, and it is named as insulin non-dependent diabetes mellitus, or type 2, that in this time, insulin secretion is not impaired, even it is higher than normal, but insulin is not able to exert its effects. About the cows, I can say that type 1 diabetes, that is juvenile onset diabetes, mainly is familial. Sometimes it is autoimmune disease, sometimes viral infection, and mutation in the genes may cause such problem. But sometimes, but for diabetes type 2, mainly nutrition, lifestyle, metabolic disorder, and sometimes genetic factors also. will cause, and this type will develop even in the later of life. For example, we know obesity, inflammation, continuous inflammation will phosphorylate the IRS receptor, IRS substrate, and inhibit its activity, and it inhibit the insulin receptor activity. I'm not going to discuss about the disease. I just want to show you how much insulin was important. You know that diabetes is known by some clinical presentations such as hyperglycemia, that you know why, because insulin is not able to stimulate glucose uptake. Glucosuria, appearance of glucose in the urine, because high glucose is the kidney is not able to uptake all the glucose from the urine. It is saturated, and therefore glucose will appear in the urine. One reason that we say that the diabetes mellitus is the sweet taste that they have in the urine, and historically it was known that those who have diabetes mellitus, when they pee in some area, they will attract insects. Polyphagia, because of the, in fact, I can say that cells are hungry even in the presence of high glucose. Polydipsia, eating, drinking water, because they are all the time they are losing water from the urine, and some other symptoms that you can see here. One clinical test, as a final slide, one clinical test that we can measure the insulin resistance is named as oral glucose tolerance test, OGTT. And you know, this is very easy. Firstly, we measure the glucose and insulin concentration in a fasting state, and then ask the patient to drink a high glucose content. In normal situation, this causes the glucose after some minute, the glucose concentration to increase, and then decrease in the glucose concentration will be seen. But in diabetic patient, you can see that this will increase the glucose concentration and reach very high levels. When we, at the same time, we measure glucose concentration and insulin concentration, sometimes we have a person that have a normal OGTT, but we see that this returning of glucose to normal is causing a high level of insulin secretion, and this shows that we have a level of resistance against the insulin. By this slide, and this is showing the treatment such as type 1 that we inject insulin, and type 2 that dieting, exercise, drugs to increase sensitivity, drugs to increase release will be used. By this slide, I am going to finish this part and also the subject of pancreatic hormones. In next files, I will continue the endocrine physiology with the hormones responsible for controlling calcium and phosphate concentration. And finally, we will go to reproductive system and hormone of reproductive system. Thank you very much for being with me in this subject. Have a good time. Take care.